Intrauterine growth restriction, or IUGR, affects 10 to 15% of pregnancies worldwide. Compared to infants who are born small for gestational age, IUGR is pathological in that it is associated with an increased incidence of perinatal morbidity and mortality. A common etiology of IUGR is placental insufficiency, which is defined as the inadequate delivery of blood flow and subsequently oxygen and nutrients to the developing fetus. When the placenta fails to deliver adequate oxygen during placental insufficiency IUGR, a hypoxic growth environment evolves. Subsequently, damaging oxidative stress, described as the imbalance between reactive oxygen species, or ROS for short, and endogenous antioxidant mechanisms occurs. As raw surface signaling molecules, in addition to promoting cellular damage, oxidative stress is thought to promote a cascade of fetal reprogramming within the developing fetus that results in gene expression changes with negative physiological outcomes. The hypoxic growth environment and eventual oxidative stress is especially prominent within developing skeletal muscle during IUGR. During placental insufficiency IUGR, blood is shunted to vital organs for eventual postnatal survival, including the brain and the heart, while diminished blood flow reaches the skeletal muscle as a result. A reduction in skeletal muscle mass and infant size is evident, in part due to the decrease in organ and nutrients delivered to the developing organ, but it has also been proposed that oxidative stress induces inhibitory molecular changes to myogenesis, i.e., the process of skeletal muscle development. Similarly, muscle mitochondrial dysfunction is associated with IUGR, and oxidative stress is proposed to induce this defect as well. Understanding how oxidative stress impairs regulation of myogenesis and muscle mitochondrial function during IUGR is an important issue to address, as both appear to predispose the IUGR infant to later life development of non-communicable diseases, such as type 2 diabetes and cardiovascular disease. And the prevalence of such disorders is increasing worldwide. To prevent oxidative stress and resulting metabolic consequences in IUGR, antioxidant supplementation has been proposed to outcompete the increased ROS concentrations present. PQQ has been specifically suggested, as it has been shown to not only decrease oxidative stress, but also increase expression of mitochondrial genes and subsequently the function of the organelle, including increasing the expression of genes specifically shown to be downregulated during IUGR and likely contributing factors to the muscle mitochondrial dysfunction seen. After exposure to developing skeletal muscle in vitro and in vivo during oxidative stress and IUGR respectively, the studies determined that in utero exposure does not increase expression of mitochondrial genes in the skeletal muscle as predicted, and appears instead to inhibit gene expression of those associated with myogenesis too. In other words, PQQ appears to augment the effects of in utero oxidative stress, and its administration during pregnancy should be reevaluated and further studied.